Hello, everyone. Welcome to this year's last M2D2 for summer break. So today we have Chaitanya, who is a PhD student at the University of Cambridge, supervised by Pietro Leo. He's working on graph neural networks and geometric deep learning for biomolecular modeling and design, and he has had a recent interest in RNA 3D structure and structure-based RNA design, which I think we'll hear about today.
Yeah, so thanks for the invitation. So RNA molecules have been at the forefront of biology. I'm not a biologist, right? I'm a computer scientist by background, and these were just some of the books I was reading at the start of my PhD and around when the pandemic was ending. And talking to biologists, I realized like there's a lot of excitement for RNA these days. Um, and that's what really motivated me to look at these things in my PhD. Um, and the other thing that seems to be an emerging picture in biology is that many RNA are structured, unlike what people may know previously. Many RNAs have function due to their structure. There's a really nice talk by Riju Das on this topic, which I highly recommend. And meanwhile, right, generative models are taking over protein design. And there have been many prominent papers, and I sort of saw this with my own eyes at Prescient Design in Genentech during an internship. There's even been $1 billion startup on this topic, right? So the question is, how do we bring to bear these tools for designing RNA? And that's what my talk is about today. I'm Chaitanya Joshi, and this is joint work with lots of people from Pietro Leo's lab. Um, and the title of this talk is gRNAde, Geometric Deep Learning for 3D RNA Design. And the paper is online, and we've also made an effort to like release code and make some of this accessible to people. So before I start the talk, right, I like to do a 1 or 2 minute summary slide. And if you tune out after this, hopefully you still get something from the talk. Um, gRNAde is this tool for inverse design, which is the problem of taking a backbone 3D structure, so a structural scaffold, which roughly accommodates the functions that you want your molecule to have, and design a sequence for it. And within this black box, we use a graph neural network to encode structure and a language model to decode sequences. And something that's new in our work is that we actually don't just consider a single structure, we actually consider multiple structures, multiple states, because this is often important for designing RNAs. We want the same sequence to accommodate multiple states. And our graph neural networks are modeling all the symmetries that we need in order to model the 3D structure and the multi-state nature of RNAs. And we feed those encodings into a decoder to design or generate RNA sequences, which the model is essentially trained to generate sequences which it thinks are likely to fold back into a certain backbone. In terms of our main empirical results, right, we compare gRNAde to Rosetta, which is the previous state-of-the-art physics-based tool for RNA inverse design in 3D. This tool was developed a decade ago, but we were happy to see that our model is performing better in general, but more importantly, is significantly faster than physics-based tools like Rosetta. Which we think makes it a lot more broadly accessible and perhaps makes it easier for people to use these new tools for designing biomolecules. And we're really inspired by things such as protein MPNN, if you're familiar with that, for protein design. And we've tried to make this easy for people to use. So we've tried to release the code and collab such that people can reproduce and perhaps use this tool in their own RNA design scenarios. So, I also recommend people to just check out the collab and play with it as well. So that's for summary, right? And by the way, I'm happy to be interrupted for questions if people have them during the talk as well. So before I dive in, right, I want to give a bit of background and like set the stage a little bit about why care about RNA when, so much exciting things are happening in like protein modeling and design with deep learning, right? Like every week there's like some big paper on it these days. So what we learn when we learn molecular biology is this central dogma. Proteins carry out all cellular functions. DNA is the genetic code, and RNA is this temporary messenger that sort of turns DNA into proteins. So when we fall ill, then something goes wrong with proteins. Um, and conventional therapeutics target proteins. They try to bind to proteins in the cell, and this would perhaps destroy or make certain proteins inactive, and then you'd be healthy again. And, antibodies and small molecules are roughly working this way, right? Um, and these therapeutics are treating symptoms. So symptoms when they appear, and they're countering end stages of disease. And you know, so essentially, you take these therapeutics when you see disease symptoms and when something goes wrong with proteins in your body. What's emerging in biology is this dual role of RNA as a carrier genetic information and performing function or having a functional role due to its structure. And that's led to a rise in RNA therapeutics, which are essentially intervening much earlier in this pipeline, okay? So with RNA, you can control which proteins are being expressed in the cell. And a lot of RNA therapeutics are functioning upon this idea that if you know about disease-causing information pipelines in the cell, you can perhaps cut off that information much earlier through RNAs, and intervene much earlier. And that's led to a lot of excitement in, of course, mRNA therapeutics, mRNA vaccines, perhaps even vaccines for cancer, but also other kinds of RNA-based therapeutics. So that's what I'm excited about as well. And, thinking about how machine learning tools could be perhaps useful for the development of this kind of biomedicine. But then, when we think about what's happening in AI and in deep learning, right? Generative AI, which is a word I don't like, is really starting to work for protein design, right? And here's a rough pipeline for protein design with deep learning. One of the assumptions you make is that structure determines function. So if you can design particular structures, then you can get the same function. So perhaps you'd start with a structure that you know has the function you want from the protein data bank. And then what you do is you extract a skeleton or a scaffold or a backbone from it. So you strip away the identity of the residues, and you only keep the backbone. And then you inverse fold it. So you use tools such as protein MPNN to predict or design sequences that are likely to fold into that backbone. And this is what these models are trained for. And these are your designs, like these are your designed proteins, which you can then actually computationally validate using a structure predictor. So now you can map from your design sequences to their predicted structures and verify whether they fold back. So for instance, you could use AlphaFold to do this. And this pipeline's been pretty successful, especially with these new deep learning tools. There's a lot of excitement in using this pipeline. And, of course, the assumption here is that structure determines function. What's not shown is that there are also purely sequence-based approaches, protein language models, which are also being used for designing biomolecules as well. So a simple analogy here is to ChatGPT, right? And natural language models are often conditioned on some prompt and then predicting the next token. Well, in the case of proteins, right, what Ingram et al. Proposed a long time ago was structure conditioned sequence generation, almost like structure informed language models, which are essentially prompted with a 3D backbone structure, and then you're generating the language of proteins or of RNA or of other biomolecules. And these models have, recently been shown to be highly effective for protein design. And, we tried to build the analog for RNA essentially. Now, one of the things to mention here is that these structure conditioned language models are trained on PDB structures. So they're, of course, going to be biased towards what's in the PDB. So they're biased to sequences that are likely to be thermally stable, for instance, which could be a good or bad thing for your design. The other paradigm, which I'll touch on briefly, right, is that you're actually starting from scratch, de novo. And here the main tool is called RF diffusion, which is a model to start from some target or constraints and sample new backbones. So generate novel protein scaffolds in order to design them. And this approach is also hugely exciting, but not what this talk is about. For backbone design, the analogy really is to DALI type models. They all use the diffusion framework to sort of learn to map from noisy samples to clean samples and to learn data distributions this way. So these were just some analogies and some background, right, on this emerging field. And that brings us to thinking about RNA. Like, why hasn't this been done, and why is it difficult, and how we went about starting to do some of this work? Well, when people think about RNA, they actually think mostly about its secondary structure, especially for design. There's interest in RNA tertiary structure prediction, but as this article that I've referenced here by Vincent's says, right, actually a lot of interesting functionality in RNAs, especially in structured RNAs, comes from the kinds of interactions that secondary structure never captures, non-canonical interactions and sort of interactions due to tertiary motifs, which are not represented in secondary structure at all. So there is interest in doing design in 3D, but there are relatively fewer tools. There are a few potential applications, essentially anywhere where structured RNAs come up. Two that I'm interested in are as follows, right? The first one is essentially transient gene expression, where essentially you can, if you have an mRNA which is expressing a protein, you can design on-off switches, or we dream of designing on-off switches for such RNAs. So we need to design structured RNA elements that we can essentially program into switches with logic in them, such that we can have extremely targeted therapeutics. And then the other application I'm quite interested in is ribozymes, which are related to this hypothesis called the RNA world, where it's hypothesized that life began as RNA and then later evolved to have proteins and DNA. And so then something that people are interested in is like, what is the diversity of RNA sequences that can actually be functional and carry out functionality? And in both these applications, RNAs, they carry information, they carry the genetic code, but they also play functional roles. And it's really interesting, like how diverse RNA can be. And these are just some of the applications that we might want to design RNA from a tertiary perspective for. So one such tool, right, I just wanted to talk about was RNA-Make. And this is a tool which uses classical algorithms to align RNA Lego blocks, essentially like small motifs that they're aligning and stitching together using classical search algorithms. And they've shown that for specific design tasks, you can actually build realistic RNA, which actually work. Like they've experimentally validated this. And, they've shown that it can work, for instance, for locking aptamers into their bound state, which improves their binding affinity. One limitation of this sort of older class of models or tools could be that they're based on heuristics which are, hand engineered. So they may not be generally applicable, or, it may be interesting to learn RNA design heuristics in a data-driven fashion, which our work and others' work is sort of building towards in what I really see as like a emerging sort of deep learning based toolkit for RNA design. So this talk is about gRNAde, which is our equivalent of protein MPNN, so the inverse folding part of this pipeline. But we know that several teams are actually working on AlphaFold for RNA. Well, there's the new AlphaFold, which does RNA as well, right? And based on its performance, we also know that this is a really difficult problem, right? But several people are working on building better RNA 3D structure predictors. And of course, RNA language models as well. So Rinalmo is a recent, pretty nice RNA language model similar to ESM. And then there's nothing public yet on the sort of generative design side, although I might talk about this at the end of the talk. So let's start thinking about deep learning, right? Like when we think about, well, we want to build deep learning tools, the first question is what data exists? And the answer is not that much. The main challenge really is the paucity of data. There is about 12,000 RNA structures from around 4,000 unique RNA. So there is some amount of redundancy in this data set in the PDB. But when you compare that to known protein structures, there's around 200,000 of them from 25,000 representatives. So like an order of magnitude difference, right? And this is reflected in just essentially far less machine learning people then work on RNA, right? And nobody's like bothered to process this data well and think about this problem deeply. In fact, when we look at some prominent papers, you see that the amount of data that they use is really few. So in our work, we went and collected this RNA 3D structure data. We actually used RNA Solo, which is a nice repository which collects these structures from solo RNAs, but also from complexes. So it cuts out RNAs from complexes, which can be tricky because if you're cutting molecules out of complexes, they're obviously in their bound form, but they can still teach you something about the sort of way that RNAs are forming structures. And so we took this data set called RNA Solo. Of course, it's one order of magnitude below what's available for proteins, but we tried to process this data and sort of release it in a way that others could use it as well. And we're building our work upon what's available out there, but it's also natural to ask, like, should we just wait and should we wait for the PDB of RNA to get populated? And there is some excitement for this because it seems like we can use cryo-EM to solve some RNA structures, but obviously that's going to take a lot of time. And I think it may not necessarily be the case that we need to just wait for the PDB to be saturated enough, because we have seen that there are cases where you could think more carefully about your training data. and still build useful tools, which is often relevant because you actually want your tools to be used a lot. Like the inference speed for tools should be fast. So it may be interesting to think about building tools with the data that we currently have and see, to what extent they are actually working. So then we started looking at this, right? And here's just like a couple of slides on what data is in there. Um, Here's just the plot of like frequency of sequence lengths. And, you can see that there are like these distinct clusters, which actually are you mainly coming from like specific families or types of RNAs. Our RNAs are ribosomal RNAs, which have very specific lengths. Um, and the ones that we're probably interested in are like shorter structured RNA, where there is some amount of diversity, although over there as well, there is like overrepresentation of certain classes of RNAs. Um, the other thing about this data set, besides sequence length, is that many of these RNA sequences often come with multiple structures. So for the same RNA, you often have multiple 3D structures, which can actually be quite different as well, depending on how you measure things. Um, and, one way to think about this is that it could be that multiple conformations are important for the functionality of RNA. And for many applications, we do want RNA to have multiple states or multiple conformations. So another feature of like the data that we were collecting was just being careful or having a look at like um, those RNAs where you have multiple structures, trying to measure like how diverse are the structures. And we do see that some sequences can have pretty diverse set of structures available for them. We also tried to see whether this effect is simply due to sequence length, but we find that that's actually not the case, and that like many shorter RNAs also have multiple states available for them. Um, the other reason why this could happen could also be that it's harder to um, it's harder to determine the structure of RNA. So it might be that they're being determined in multiple states as well. And then the last thing, I guess, about this data set is that it is biased. Like it's very biased towards ribosomal RNA, which are quite special because they actually form their structure due to um, interactions with very specific proteins in the ribosome. Um, but we stood to reason that even from really large ribosomal data, when you look at substructures and local motifs, you could still learn something about RNA biochemistry and RNA structure. Um, and essentially, like the class of models that we use, like graph neural networks, these are localized models. So maybe if things are locally still telling you something about RNA structure, you can still learn from them. In our inverse folding problem. Of course, this bias could be a big problem for structure prediction, which is not really a local problem, right? That's a more global problem where, if your data set is biased to certain global folds, maybe your model will get really good at those folds, but have a hard time potentially generalizing as well. So that was for the data. When we when we start thinking about now what we've built, we call it gRNAde, and it's a pipeline for inverse folding or fixed backbone redesign. It's taking as input a PDB file and it's outputting designed sequences, with the idea being that you can sample many sequences for your backbone and essentially look at a quasi-species population, or you can look at what is the diversity of sequences that could form your tertiary structure or your tertiary motifs. And this system is trained in a self-supervised manner. So all you need are those PDB structures because your input is the backbone that you get from the 3D coordinates, and your output is the sequence. And you can essentially train these models without, let's say, like experimental um, labels, although the coordinates are obviously determined by experiments as well. So to build this pipeline, right, like the first step that you need to do is you need to featureize backbones as 3D graphs. So you have to take your PDB files and process them into these graph structures. So the analog of residues in proteins is called nucleotides for RNA. And these are like units in your structure. So units or groups of atoms. And they're composed of backbone, this sort of backbone called a phosphate group here, a ribose sugar, which is a 5-membered ring, and then base atoms. Bases are essentially like side chains for proteins, and the identity of the base atoms is what's not known in this case. And we have the coordinates of the backbone atoms, but not of the base, and our model needs to predict this. So when we process our data, we retain a few key atoms in our nucleotide. So we retain the phosphate, the carbon of the ribose ring, and then the first nitrogen that's attached to the base. And retaining these atoms doesn't give the model, like it doesn't leak information about the task, right, which is to predict the base. But it's a coarse graining. So it's a sort of way of reducing the number of degrees of freedom that you might have for your input features. And this is for a single nucleotide, a single residue, let's say. And each residue essentially becomes a node in your 3D graph. And you connect these nodes to their nearest neighbors in 3D space. So you may look at like your k nearest neighbors in 3D space and connect them via edges. And each node's features are essentially some local coordinates, some internal coordinates for each nucleotide. So each nucleotide essentially is, some geometric features like distances, angles, and torsions locally within the nucleotide's atoms. And the task is to predict the identity of the base, which we would like to design. So essentially, given this 3D graph stripped off of the identities of the bases, you want to predict the entry for each node, or predict the nucleotide, the base for each node. Now, here's just one slide on like why we chose the 3-bead representation. There's some really interesting work from Anna Pyle's lab at Yale showing that even though RNA backbones have a lot of torsion angles, right? So in principle, they could be extremely flexible and malleable. They found that you can describe almost all the configurations of these backbones using these two pseudo torsions. So that's a way to, prevent your model from perhaps overfitting to artifacts in your data, or noise in your data, and potentially making it more robust, just based on some of the literature. So that's how you prepare your graphs, right? You featureized your graphs, and if you have more than one backbone structure, you'd actually have featureized multiple graphs. And then the idea is that for every node in your graph, you now need to predict the probability over 4 possible bases. So these are A, T, G, and A, U, G, and C, which are like the 4 bases in RNA. So you start with these backbone graphs, and we use essentially these SE3 equivariant GNN layers, GVP GNNs in our case, to encode each of the backbones. So essentially for every node in every backbone, you're learning a node embedding through multiple GVP GNN layers. And the model is sort of Yeah, you can go ahead and ask your question as well.
Oh, sorry, there's a question in the chat asking
Yeah.
if RNA structure prediction is harder because there are only 4 nucleotides as
Mhm.
opposed to the 20 amino acids in the proteins. Does that make sense?
Yeah, I mean, I think RNA structure prediction is harder because there's less data for it primarily, right? But it it might be harder because of the fact that RNAs seem to be conformationally more dynamic than proteins. Like there might be less data for it because it's harder to determine that data, right? So I think I think that's the reason why RNA structure prediction is harder. Like RNAs are conformationally more dynamic, and that means that you have fewer for them. Why are RNAs conformationally more dynamic? Um, I don't know if I know the answer to this as well, or like we fully understand the biology or the biochemistry here. I think one of the reasons people cite is that the RNA backbone, like the thing that I showed, it has these phosphate groups, which are phosphorus and oxygen atoms, and that like these are highly negatively charged groups. So this like this negative charge on the RNA backbone is different from protein backbones, which are actually neutral, and maybe the negative charge leads to the RNA sort of dancing around more. I may have gotten that completely wrong, but because like I haven't trained in this, but this is just something that I've sort of understood from reading things. But
Yeah.
yeah, I mean, there could be interesting sort of chemical reasons why RNA are conformationally more diverse and hence harder to collect data for as well. Um, yeah.
Yeah, there's 2 more related questions in the chat. That's okay.
Mhm.
The
Yeah.
first is asking, why do we have multiple backbones for each RNA?
Yeah. So, I mean, that's a good question, right? And um, the reason is that when we want to do design, right, we usually want to design for certain function, right? And in the case of RNA, when I was reading about this, right, it seemed like the whole field was really excited about how RNA structures as conformational propensity determines functionality. So, if you're able to modulate your RNA to occupy, say, two specific structural states or switch between them. For instance, like if your RNA binds to some ligand, some other molecule, and switches to a different state, right? Um, People use these kind of mechanisms to design function or to think about RNA function. In fact, like we know of such RNA molecules. These are called riboswitches, and these are found in bacteria. So essentially, like bacterial RNA are sensing certain small molecules and then turning on, and that will then express some protein downstream. So we wanted to incorporate this multi-state thing into our model because we thought like, if we really want to use this for interesting biotechnology applications, then we really want to design this switching of structural states. And obviously, the same ideas, the same kind of principles also apply to proteins and protein design. And, like we're sort of exploring these ideas for proteins as well. Yeah.
Yeah,
I
thanks.
can,
There's more questions. I think you kind of answered them, but I can
Mhm.
read them if you want.
yeah, I can
So,
read through
oh,
them
you
as
can read them as well.
Yeah.
Yeah.
So Carl asks if your model receives any features to know whether the RNA was incomplex. So not yet, right? But I think this would be like something we actually do want to build into the model, right? Like being able to condition based on partners, right? Is something we want to do. And Zoeb asks, are there epigenetic factors impacting RNA structure like methylation, say? So I think there are actually. I went to an RNA conference recently, and they actually showed that there are even differences in the structure of the same RNA the longer it stays in the cell. So, like that tells us actually how little we know about like even the basic biology of these things, and how maybe, like there are so many factors that we're not really considering in biomolecule design, or we have no way of like really considering. But the fact that the structure of these systems actually changes as they stay in the cell, and probably all the PDB structures we use are just models or inductive biases, and maybe like what really matters is the sequences. um, so yeah, I think structure does change a lot, and that it's quite tricky to actually model these kinds of structures. Um, but here's how we do it, right? So we have multiple states because we want to perhaps design functions which are due to multiple states, and we encode each state with a GNN. Um, and here, like, of course, like these GNNs are, equivariant to 3D rotations. I'll talk about that soon. And they independently encode each of the backbones. But doing this in, say, PyTorch geometric can be quite inefficient. So what we did was we tried to unify this. So in terms of implementation, actually doing this kind of GNN layer over multiple independent backbones, we tried to put it together into a single layer, which made it a lot faster and memory efficient for us. And we called it a multi-state GNN. Um, and so once you get these encodings or embeddings from each state, you want to pool them together. And we do something really simple, like we just average together the embeddings of multiple states, or we use deep sets essentially to sum them together. Um, and then you want to decode the sequence for each nucleotide. So you'd pass this to an autoregressive decoder, which per node, it produces some logits, some probabilities, from which you sample and select the identity of the base. And like you do this step repeatedly to design your sequence in an order which goes from, say, like the 5 prime, the leftmost end of your RNA, to the 3 prime, the rightmost end of your RNA. So that's the pipeline, and it's essentially following best practices in protein design. So protein MPNN and similar tools are using pretty much the same pipeline. And our main contribution was porting this over to RNA and actually trying to build a multi-state version of this pipeline, which we thought was quite specific to RNA. But obviously, we're also interested in using for proteins as well. So this is for the pipeline. Carl asks another question, that does the autoregressive decoder get the sequence only or the probabilities as well? So that's an interesting question. I think like that could be quite interesting to try. Like currently it only gets the partial sequence, but yeah, I mean, there could be ways that you could like feed in the predicted logits as well of your model. To sort of indicate the relative ranking of different things. But I think like one of the reasons why like things get the partial sequence is that, if you think about RNA structure, a lot of it is about base pairing. So you'd have two strands, and then you'd have like two different bases across the two strands, which have hydrogen bonds across them. And, so if you predict something for this particular strand, then you need to predict the exact complementary base for the other strand. And, that way you kind of perhaps have far fewer choices. Like if you've predicted something for this strand, then like that almost automatically determines what you might predict for the next strand if you fix the base pairing. So that might be interesting though to consider this. So that was for like our pipeline, like that was for the machine learning sort of model, which takes as input structure and decodes sequences, right? You give it a backbone graph, you get sequences. How do you then choose what are good designs, right? How do you evaluate the system, and how do you perhaps select like what gets sent to the lab? The one I'll show you today mostly is called sequence recovery. So you can basically measure how well you recovered the true sequence in your designed sequences. Um, of course, this is not a perfect metric, right? It's the most popular one in the community, but it's really not perfect in the sense that you don't want to be really good at recovery. You want to be sufficiently good, but you actually want diverse sequences different from your true sequence. So one other metric is to perhaps take your design sequences and fold them using a structure predictor. And in the case of RNA, we can consider 2D and 3D structure prediction. And then compute how well you recover structure. So you can compute structural self-consistency metrics. Um, Now, there's a big caveat to these metrics in the sense that RNA structure prediction is not as reliable as protein structure, especially 3D structure prediction. So that means that, if your structure prediction tool is bad for certain types of RNA, then it might be the case that your model would not perform very well for those either. But this is generally, a framework that we use for evaluating. So a lot of comparisons are made using sequence recovery, but perhaps like when we're now actually using our model, right, for real design scenarios, we actually care more about structural recovery. Here's a short aside on graph neural networks for 3D structure, but I won't spend time on this today, actually. We've written a consensus article with a few different groups on this class of models, which we think are, really the core machine learning tool that's driving a lot of progress across areas of biology and chemistry. So if people are interested, then I think this is a good starting point. Sean asks, how did you process cases where multiple RNA sequences share the same backbone, if there are any? So um, in this case, right, we just treated them as different samples. So our definition of sample was unique RNA sequence. But you're right, like there could be cases where multiple sequences share the same or very similar backbones. It's just that that's also what our model is designed to do. It's designed to generate sequences that fold back into the same backbone. Um, so, like we didn't treat these samples as different in this case. Or we did treat these samples as different in this case. And I think it just follows from like how we define the problem. But, perhaps if you're doing a different problem like structure prediction, then it's quite interesting, like how you may want to treat these kinds of samples as well. So finally, let me get to like what we can do with our model, right? Maybe I'll be a bit quick. Um, so this I already briefly talked about, right? But we wanted to first start out by benchmarking just single state design and comparing to Rosetta, which was in an old paper by Riju Das and David Baker from the 2010s. They created a benchmark of 14 structured RNAs, which they were really interested in as design targets. So, we tried to compare very fairly to their benchmark, and we were happy to find that gRNAde was actually boosting sequence recovery over Rosetta and previous generation of tools by a significant amount. And, you know, what was perhaps more important was that this is much faster. Like the deep learning based approach is just you train your model, it takes 5 or 6 hours, but after that you can do designs in under a second, okay? With physics-based tools, every single design that you do needs to run a sort of search algorithm, an MCMC sort of sampling procedure, and this can take hours. And the Rosetta documentation itself says that to design anything with hundreds of nucleotides, it could take hours because maybe this is not yet optimized. Maybe that it could be, but it's still like it's not as broadly usable as deep learning tools are. And here, like just to mention that we did try to be as fair as we could. And, we actually excluded all homologous and structurally similar RNAs to those that we evaluate on, so that we're evaluating our deep learning model on generalization as much as possible. So the model does better than like previous generation of tools, right? But that could also be because people haven't really been working on these tools. But we still thought that it's sort of a way to like restart interest in like these sort of approaches, or make these approaches more useful. Now, some of the other things I want to show, right, is this is the same benchmark of 14 RNAs, and I'm plotting Rosetta versus gRNAde recovery. And what we see here is that perplexity, this is in addition to designing a sequence, right? You can compute the likelihood of the design using gRNAde, since it's a language model. So lower perplexity is more confidence, let's say. gRNAde feels more confident about its design. And what we are showing here is that perplexity seems somewhat correlated with recovery of sequence. And of course, this depends on different targets, right? So like for some targets, gRNAde is really good, and for others, maybe Rosetta is better, and for others, maybe it's challenging for both of them. But generally, we were seeing that gRNAde's perplexity is correlated with recovering the ground truth sequence, which was promising. So then when we took these designs and then we predicted their 3D structure, we were sort of again able to see that perplexity from gRNAde seems to be correlated with how well you recover structure as well. So here I'm showing some cherry pick designs, right? Sort of in the order of like better to worse perplexity. And then we're computing some of these self-consistency metrics for 3D structure. And we were generally seeing that like designs with lower perplexity are actually having better 3D metrics as well. Of course, this is cherry picked, but here, like just to show you like the range of like how the model does, right? For some RNAs, it could be really good, and over there, the trend is more clear that like when you have a high perplexity, it's really likely that you get a good SCTM score, and SCTM means like self-consistency TM, wherein this TM score is just a metric for measuring how similar two RNA structures are. And essentially, like, we saw that many of the designs actually have the same global fold as your as the structure that you do want to design for. But for other RNAs, it may not be so great, but you could still see the trend. And then there could also be scenarios where the model or the structure prediction tool just completely fail, or don't give us satisfying results. But this is just to show that there are cases where the model is good at, and there are cases where the model is limited as well. So the main point here was it seems like the model's learning something about RNA structure, right? Like its perplexity, its inner sort of notion of confidence is correlated with sequence and with structure recovery. And then the question is like, could we also be correlated with fitness or function? Like that would be really interesting, because we obviously want to design for function. Yeah, we went to see if like can gRNAde understand fitness, right? And here we did a sort of retrospective analysis on data from Phil Holliger's lab at the MRC-LMB in Cambridge. They've released a cryo-EM structure of an RNA polymerase ribozyme. This is self-replicating ribozyme, an RNA that has catalytic activity. And this was not in our training data. But what's really cool about this study is that with their structure, they've actually released fitness measurements through a very high throughput fitness assay of 75,000 mutant sequences. So you make some random changes in your original sequence and you measure how the fitness or function of your RNA actually differs now. And this data is really interesting because we can now use gRNAde's perplexity as a way to rank sequences. So gRNAde's perplexity is the likelihood of a sequence forming a backbone, and we can then use it to rank these 75,000 mutants. And we can see how well gRNAde understands which mutations perhaps destroy the RNA structure and hence destroy its function. And get like some initial signals on whether this tool is potentially useful for designing functional RNAs. So here I'll like show very quickly this retrospective study that we did. So on the y-axis, right, is expected maximum change in fitness, because this is the goal of RNA engineering. You want to start with an RNA and you want to improve its fitness. And on the x-axis is the number of sequences that you choose to send to your wet lab. Or, we were like retrospectively simulating this. So like a sort of simulation of how many sequences are being evaluated. And we'll also plot, say, like the absolute fitness values on the right side. And you can see, for instance, right, like that the 50th best mutant in the data set would have a fourfold improvement in fitness, for instance. And what we'll plot, right, is what is the best fitness improvement if I were to select, say, 10 or 50 or 100 designs at random versus using gRNAde. And then like the other two here are at random from all first order mutants, so making only one mutation, or at random from all up to second order mutants, so making up to two mutations in the sequence. And these are like very simple strategies used by people in RNA engineering. So we wanted to see if, using gRNAde can be useful for picking good mutants. So we ran these kind of simulations, right? And what I'm showing here is that for these extremely low throughput regimes, it seems that gRNAde's perplexity is able to actually rank and identify good mutants much sooner than random. So it takes much fewer sequences for you to find good fit mutants compared to selecting them at random. Which was quite promising. Even when you exhaust all possible single mutants, we saw that gRNAde was finding better mutants. And as you sort of randomly choose more mutants, of course, then you start doing better and better. But we were still, interested to see, and this seemed like there were some signs of life in this sort of approach, that like our model was able to identify certain good mutants for this ribozyme, right? So now we're trying to actually experimentally validate this tool as well. And that brings me to multi-state design. I think I briefly touched on this, and I think I'll like almost skip through this actually. But we want to design RNAs that can undergo conformational changes, and nobody's like built a benchmark for this before. So we set out to build one, and what we were seeing is that multi-state models, like models which are explicitly multi-state versus the sort of single-state approach, were actually improving recovery very slightly. We hypothesized that this may be due to improvements in flexible regions. And, here, for instance, like something that I could show very quickly is that when we did look at per nucleotide sequence recoveries and measured how flexible that nucleotide is, then we were actually able to see differences where the single state model would actually be worse at nucleotides that were in very flexible regions of the RNA, which we thought was, again, like promising. And, we're really interested in actually seeing if this multi-state approach works. So I'll conclude, right, with that, some of the things we're thinking about and some of the limitations of this work, right? We really want to use this, or like we want to find out if this approach works. Like we don't just want to like benchmark it in an ML way, but we're interested in helping people to actually use it. So we are exploring some applications as well. I think to make this tool more broadly useful, we want to actually support like interaction partners. That's something we touched on today as well. And of course, we want to release these resources as tools that people can really use. And we've been doing some of that. So, I recommend people to check out our code base and, perhaps, if you have some RNAs of interest, you can actually use gRNAde on Colab on your own RNAs, and you'd get a sort of dashboard where you can visualize your designs, and you even get their PDB files, which you can then view their structures using. So we want to make these resources more useful for people and see if these things really work. And then like something to just end off, like something new that we're doing is actually trying to design novel structural elements with Rishabh Anand, the really talented undergrad from NUS. We've we've tried to see how to what extent we can push tools from protein design and see whether they work for RNA. And we've been able to get somewhere. We've been seeing that our models are able to generate backbone 3D structures that are valid over 40% of the time. But it's also really challenging, and like something that we do say in the paper, which hopefully should be an archive soon, is that we feel like these models are quite expressive, like they can learn training distributions. But in the case of RNA, if your training distribution is quite biased towards certain structural families, then your generative models really kind of latch on to those families and get really good at almost regurgitating the training distribution. But that may not be the goal of design. And it seems like designing truly novel structures is still, something that needs more work. And we want to, enable more people to do that work as well. Um, Another sort of notable place where gRNAde was being used is in a recent paper by MIT on RNA flow, very similarly named to our work, which also uses flow matching and gRNAde with Rosetta fold NA to do sort of protein conditioned sequence generation. So with that, like thanks to my lab mates and people who encouraged me, and thank you for listening. And I'm really happy to connect with people as well.
Awesome. Thank you for your talk. I guess we're a bit over time. If people have short questions, we can them. Otherwise, oh yeah, seems to be some questions. All right.
Yeah, I mean, happy to chat offline as well. And like, yeah, I mean, people should feel free to like ask questions as well.
Yeah, Ravi, if you want to go
I
ahead.
can ask my questions. Is that fine? Or
Yeah.
thank you, Chaitanya. I just want to ask a few quick questions. I know we're over time. Maybe I'll ask up to two. In your presentation, Chaitanya, I know I could be wrong, but I know for a fact that when you on a few of your slides you briefly touched on the graph neural networks. So I just wanted to ask about that. Um, did you by any chance compare test graph neural GNNs or graph neural networks on gRNAde or any such variants? And if so, did you compare
Yeah.
any multiple types of GNNs? That is my first
Yeah.
question. And
Yeah.
my second question is, it might be a bit irrelevant, but I'll ask anyway. Have you tried like experimenting with, like, say, RNA mutations, so to speak? Those are my two questions. Thank you.
Yeah, I mean, so obviously, like in my other sort of PhD research, I've been thinking a lot about like GNN architectures for 3D structure, and I did try to ablate this, right? Um, it's really interesting that it seems like it doesn't matter too much for this problem. I think like all these models are capable enough to get good performance, roughly equal performance for this problem. So we didn't see like major improvements from using, say, more expressive models. Obviously, like that's not to say that expressive models don't have a use, right? I think depending on the problem, it may be the case that, you either need more expressive architectures to model the underlying phenomenon, or sometimes, you're actually battling the other problem of, say, overfitting, which I think is the case here. And then you perhaps want to like use more regularized architectures, which are like perhaps less expressive and don't overfit to spurious patterns in your data as well. Um, and then the other question was like, have you experimented with using mutations? Um, I mean, I think like I don't know. I think it depends like how we use them, right? Like you could use them to augment your training data. You could compare against them, right? Like you could evaluate against them. Um, we sort of did, right, with that retrospective study. I think like another thing is like we also compared to tools like Vienna RNA, which I think like also work on like making mutations and so on. But yeah, I mean, it wasn't like explicitly something that we were using, I guess. Yeah.
Okay. I just thought that if you if you tried like RNA mutations, I just thought that it would be interesting to see what kind of results we get, and how we can, and what kinds of differences are there between like different RNA structures or structures with the mutations. I just thought I just thought I just
Do you
thought
mean like
I'd ask.
modified nucleotides, like modified RNA bases, like changing the chemistry of the RNAs?
Mhm.
Because that's something we don't consider, and we should to make this work realistic, right? But it's really hard. Like even considering like modified residues in proteins is still like quite elusive, but very interesting.
If I can jump in quickly, maybe if he's talking about post-transcription mutations. So like phosphorylation and other type of mutation that can happen. But like because we are over time, maybe we can move to Zoeb for a last question and then end this.
Yeah, sure. Yeah.
Zoeb,
Anyway, those are
you
my
do still
questions.
have a
Sorry,
question?
my bad. I was
Thank you.
I muted myself. I was yapping away. There is also another question in the chat, so maybe we could get to that as well. But my question was about the data set itself that you're working with.
Yeah.
So you're doing what I think is like pretty cool work with a pretty small data set on RNA structure. At least that's what you've told us today. What do you think is sort of like your prognosis of us being able to expand that data set? Are there people working on like automatically doing this, or doing this at really high throughput or scale? Just curious what you think.
Yeah, no, I think we need to, right? Like, especially if we want to crack like structure prediction. And like I think inverse folding, right, is a very localized problem. So if you think about number of samples in inverse folding problems, perhaps it's even more sensible to think about them in terms of number of nucleotides in total. And that's quite large, right? Because like that's where your prediction is at. um, but I think like for more global problems which are like not so local, I think we do need to like bolster the training data sets, right? And there are a few efforts. So one very prominent one is called Ribonanza from Riju Das at Stanford. And they're using high throughput structure probing data. So this is like extremely high throughput experimental data that you can do to figure out something about RNA structures. So given a sequence, you can figure out, say, which positions are paired and unpaired. So quite coarse grained, but they believe that they can collect more than 1 billion sequences and such data. And they think like you can perhaps use this as a pre-training task to learn something general about RNA structure and then generalize to new problems. Um, I think there are also like people exploring to what extent we can use like physics-based tools and simulations to expand data sets, right? Which I think is like a general topic in the community of molecular modeling and something I'm really interested in as well. Um, and then the last thing is, right, like maybe what things like the recent AlphaFold and developments before that also showed is like training paradigms like diffusion and flow matching may have like some notion of implicit data augmentation or implicit regularization in them. So it could be the case that like if you trained with like noise for long enough, perhaps you could like overcome some of the data limitations. Although I think you still need a diverse data set. But yeah,
I
I mean,
feel
there are
like
efforts and there's like lots of interest in this.
I feel like, especially given what you described today, that there's a lot of value in having the much bigger data set, especially with respect to not sort of inducing some biases in the machine,
Yeah.
because so far a lot of the data has been looking at, very few cases. I think you said like ribosomal RNA was like the big.
Yeah, extremely overrepresented actually, and models do latch on to patterns from that.
Oh, of
And
course, yeah.
and like the question really is like whether the patterns and the tasks generalize to other RNAs or are super specific to ribosomal RNAs, right? And um, there's where I think like there's some interesting differences between like local and global problems.